There, my name's Emily, and I am here to talk about my uh, senior design project that I worked on with Anya. Her and I together were designed RNAs as a therapeutic for neurodegenerative diseases. We worked within the Yo Lab at UC San Diego, and our mentor was Trent Goldberg. Now, first to get into the problem, neurodegenerative diseases affects millions, but we still don't understand a lot about it. There's not a lot of genetic factors that are necessarily like contributing to the disease pathology. But what we are finding is that there might be alternative splicing events that are contributing to protein isoforms that have some sort of toxic effect. And all that complicated talk just essentially means that all of these, we have our RNA, specifically the mRNA. And it has exons, which are essentially, discrete chunks of genetic information. But sometimes specific, chunks get left in or left out. And depending on what is left in and left out will affect the eventual protein that is created. And we think that some neurodegenerative disease pathology, specifically Alzheimer's, has this phenomenon occur when a specific exon was excluded when it was supposed to be included. And that's called the tau protein. So that's sort of our proof of concept. And with that, we're trying to see if we can, try and fix that problem by making sure that certain exons remain spliced in. And so with that in mind, we want to see if we can improve splicing outcomes by engineering a more stable snRNA, which has a primary role in exon inclusion. So the snRNA is that little figure on the left. It's a molecule that comes in and binds to the mRNA, which is what that colorful little bar is in the right-hand picture. It binds in, it binds to the exon, and then says, "Hey, come here and splice here and include this exon." So with that in mind, we see that there's this fourth hairpin, circled in orange, and we wanted to play with that hairpin to see if we could, mess with its structure to see if it had any impacts on, how well it did its job. So with that in mind, we decided to design all these different hairpins. And the really cool thing about this whole project is that we were able to do a lot of our front loading our design process and creating 8 different designs and then progressing the experimentation through afterwards. Unlike most projects, which are design and then test and then redesign and test and redesign and test, we got to, kind of design in parallel, which was a really special part of this project. A lot of this stuff that we did was informed by literature and some software applications, and then to some extent, general what if curiosity. So a lot of really fun part of this design process. With that in mind, we have a pretty extensive workflow that I can't fully get into here, but involved designing and cloning, and then plasmid preparation, and then mammalian cell assay, and then flow cytometry. So back to that picture you saw, when the snRNA is able to bind and do its job, it makes sure that little pink exon is remained in, and then that turns off the TD tomato fluorescence, which, will make the cells in which this snRNA is inside, will make it turn more green. And then when the snRNA is not doing its job, we'll see more red cells. And so with those different fluorescent colored fluorescence within the cells, we can run flow cytometry to kind of gather data on how well our snRNAs are doing. And all that information led us to gathering some pretty cool data. We have a delta pseudo PSI as our percent inclusion figure, and we do see that we have many snRNA designs that are improving compared to our wild type, which is on the left-hand side. So that's a pretty exciting result. We have 2 different designs that are outperforming our wild type throughout our experiments, and it means that there is general proof of concept that this hairpin does have stabilizing effects, and that those stabilizing effects can be improved upon, perhaps for therapeutic down the line. So yeah, and there's much more to be done with all of this project. We need to continue to experiment and replicate our results, and then test within endogenous MAPT neuronal cells, and hopefully eventually develop better delivery methods so that snRNAs could be a viable therapeutic. And with that, we want to say thank you to our wonderful mentor, Trent. Unfortunately, this project will probably not be continuing for future senior design projects because our our mentor is graduating, wrapping up his PhD, but nonetheless, it was a wonderful experiment, and we have learned so much. So thank you all.